here are the different protein structures. We have the primary, secondary, tertiary, and quaternary. We also have in the right corner up here the different ways that the amino acid chains can bend depending on the types of bonds they form. So starting off with the primary, these are covalent peptide bonds. Some main components of the primary protein structure, that this is the structure that also determines the code for the entire protein. So it's basically the smallest unit of the protein that you can get. It has a basic central carbon attached to a hydrogen. There's an R group that can be basically any type of formation. There's a carboxyl group and an amino group. The primary structure is the strongest structure since it is the hardest to break out of any of them. It's the smallest and it's held together by strong covalent bonds. Okay, so now we have some different ways that the amino acids can be influenced by the type of bonds they form. So first off, we have the hydrogen bond. This is between the hydrogen and the oxygen, and it's a relatively weak bond and can be broken pretty easily. In B, we have the disulfide bridge, which is a bond between the sulfur atoms. This one is very strong since it's a covalent bond, and it's also very uncommon. In part C, we're looking at an ionic bond, which is also very strong. This is between the positive charge on the ammonia and the negative charge with the oxygen. In part D, we're looking at a van der Waals interaction. This one's pretty weak, and this is basically just a hydrophobic interaction between the CH3 bonds. And then down here, we're looking at hydrophobic exclusion. This just happens when the atoms don't bond with the water, and so they're all pushed together in this sort of lump right here. And you can see that with the CH3 again. Alright, so the next structure in the sequence of protein structures is the secondary structure. The secondary structure is defined by hydrogen bonds. Hydrogen bonds in general aren't very strong, but since there's a lot of them going on in secondary structure, it makes them stronger. The secondary structures can be separated into two basic groups, the B pleated sheet and the alpha helix. Basically, what determines whether they form as a pleated sheet or the alpha helix or in any other shape is the R group. The R group is, for instance, sticking out in a different way. The hydrogen bonds wouldn't be able to form in specific patterns. So the R group isn't directly involved in their bonds, but it can affect them. So I'll start with the B pleated sheet. As you can see, there's the hydrogen bonds. And then there's also this carbon-nitrogen backbone that kind of holds the two sections of the strands together. This bond is strong, but not as strong as the primary. And then moving on, we have the alpha helix. You can see the hydrogen bonds in this one. They're definitely set up differently, definitely due to different R groups affecting how the hydrogen bonds form. So you can see the little swirl, that's where it gets the helix name. This bond is also somewhat strong, but definitely not anywhere close to the primary structure. Okay, so moving on to the tertiary structure. The tertiary structure is defined as the final structure of a single polypeptide chain. So basically this is the final single chain that is forming a protein structure. Here you can see some secondary structures involved, and you also can see some types of bonds in here, such as ionic, hydrogen, hydrophobic, and covalent bonds. Typically, the tertiary structures are very weak compared to the primary and secondary because there's a lot going on and a lot can affect it, such as pH and temperature. And so this basically just shows the different ways that these type of chains can form. And you can just see the different kinds represented, such as the beta pleated and the alpha helix. Okay, so coming down to the last structure of the proteins, we're at the quaternary structure. So basically, the quaternary structure is defined as the final shape of two or more polypeptides. So this basically means that the tertiary is one single chain. The quaternary could be two or more of those chains that are interlaced together through different types of bonds. Just like the tertiary structure, a lot of the bonds are through the R groups, and then you can also see in here the different, as represented in the tertiary structure, the different types of bonds, such as the pleated sheets and the helixes and stuff, and the types of little bonds off to the side too. These bonds are pretty weak. They can also be taken apart pretty easily just because of the fact that there's so many different bonds going on and so many things that can affect those different bonds. Some examples of quaternary structures include hemoglobin, DNA polymers, or keratins. All right, so that is my little lesson on protein structure. I hope it was helpful.